Hello friends, in this lecture we'll talk about the carbohydrates. Here is the question which is asked in the some of the CSIR exam on carbohydrates. What kind of molecules are glucose and galactose? Anomers, epimers, conformers, and enantiomers. For this we need to know about these type of isomers that are found among the monosaccharides. Here is then another question which is asked on the carbohydrates. Which one of the following glycosaminoglycans is generally not covalently attached to protein as proteoglycans? Chondroitin sulfate, dermatan sulfate, heparin sulfate, hyaluron. So for this we need to know about the heteropolysaccharides. So let us have a look at some of the basic aspects of the carbohydrates and then we'll talk about the questions. Carbohydrates, these are polyhydroxyaldehydes or ketones or substances which yield such compounds upon hydrolysis. The empirical formula for carbohydrates is CH2ON, where N is the number of carbon. Some may also contain nitrogen, phosphorus, or sulfur. If you look at the classification of carbohydrates, these can be classified into three major classes: monosaccharides, oligosaccharides, polysaccharides. Polysaccharides can be further classified into homopolysaccharides or heteropolysaccharides. Monosaccharides, these are the simplest form of the carbohydrates which contain either aldehydes or ketones with two or more hydroxyl groups. These 6-carbon monosaccharides such as glucose and fructose, they have 5 hydroxyl groups. Many of the carbon atoms to which hydroxyl groups are attached, they are called as chiral centers, which give rise to many sugar stereoisomers found in nature. These monosaccharides, they are abundantly found in nature and most abundant among them is the sugar D-glucose, which is sometimes referred to as dextrose. Monosaccharides of more than 4 carbons may tend to have cyclic structures under aqueous conditions. Aldoses and ketoses. Aldoses are the monosaccharides with aldehyde group, whereas ketoses are the monosaccharides with keto group. On the basis of the number of carbon atoms, the further classification can be triose, tetrose, pentose, hexose, depending on the number of carbon atoms present. Accordingly, the 3-carbon sugars can be referred as aldotriose or ketotriose. 4-carbon sugars can be referred as aldotetrose or ketotetrose, and so on. Some commonly found aldoses and ketoses. The simplest monosaccharides are the 2,3-carbon trioses, glyceraldehyde, which is an aldotriose, and dihydroxyacetone, which is a ketotriose. So glyceraldehyde is a 3-carbon sugar which has at least one chiral center, whereas dihydroxyacetone is a ketotriose which doesn't have any chiral center. So let us have a look at the other sugars. The aldopentoses such as D-ribose and 2-deoxy-D-ribose, these are the components of nucleotides and nucleic acids. So difference between the ribose and deoxyribose is that there is an absence of hydroxyl group on the second position. This kind of deoxy sugars, they are found in the deoxyribonucleotides which become part of the DNA. The hexoses, which include the aldohexose like D-glucose and the ketohexose such as D-fructose. If we look at the structure, glucose is having an aldehyde group whereas fructose is having a keto group. Monosaccharides have asymmetric centers and these all the monosaccharides except dihydroxyacetone contain one or more asymmetric or chiral carbon and thus they occur in optically active isomeric forms. The simplest aldose, glyceraldehyde, contains one chiral center and therefore has two different optical isomers or enantiomers. So what are enantiomers? Enantiomers are the stereoisomers of chiral molecules in which the two forms have non-superimposable mirror images of each other. One of these two forms is designated as D isomer and the another is referred as L isomer. Here is the Fischer projection formula for glyceraldehyde shown. So here if the hydroxyl group is on the right side, it is designated as D-glyceraldehyde. If the hydroxyl group on the second carbon is on left side, it is referred as L-glyceraldehyde. So this is the structure of L-glyceraldehyde and D-glyceraldehyde. Now, in a given sugar, how many stereoisomers are possible? For that, there is one formula 2 raised to n, where n is the number of chiral centers or number of chiral carbon present in the structures. For example, in case of glyceraldehyde, there is only one chiral center, and so the number of stereoisomers possible in case of glyceraldehyde is 2. Aldohexoses, on the other hand, have 4 chiral centers, and so 16 stereoisomers are possible in this case, in the case of hexoses, aldohexoses. Examples of monosaccharides. Here, the 3 carbon sugars, 4 carbon and 5 carbon aldoses are shown. Glyceraldehyde is a aldotriose which has 1 chiral center. Erythrose has 2 chiral centers, which is tetroaldose, and aldopentose is shown over here. Ribose, arabinose, xylose are examples. Now those sugars which are shown in box, they are the sugars which are found commonly in these cells. And if you look at these aldopentoses, they have 3 chiral centers, and accordingly they can have 6 stereoisomers possible. Hexoses are shown, aldohexoses are shown, which have in all the cases 4 chiral centers are present. The glucose is shown, mannose and galactose are also shown over here, which are commonly found in the cells. These have 4 chiral centers and they can have 16 stereoisomers possible. The ketoses are shown over here. The simplest one is the dihydroxyacetone, which doesn't have any chiral center, and so no other D or L forms are possible in this case. Whereas erythrulose is a 4 carbon ketose sugar having at least one chiral center, and so 2 stereoisomers are possible in this case. Ribulose and xylulose are the examples of 5 carbon keto sugars. So if you look at the name, the UL is present in the name of these keto sugars. 6 carbon sugars are shown. Fructose is an example which has 3 chiral centers, and in this case 6 number of stereoisomers are possible. Others include tagatose, sorbose and psychose. These are examples of 6 carbon ketoses. Epimers, 2 sugars which differ only in the configuration around one carbon atom, they are called as epimers of each other. D-glucose and D-mannose, they differ at 6 C2 carbon, and so they are epimers of each other. Whereas the D-glucose and D-galactose, they differ from each other at C4 carbon and hence are called epimers of each other. Let us have a look at the structure. The glucose and mannose, they are differing with respect to the second carbon, whereas rest of the functional groups are in same orientation in both the cases. If we compare glucose and galactose, they are differing with respect to fourth carbon, and so they are epimers of each other. So glucose and mannose are epimers of each other. Glucose and galactose are epimers of each other, whereas galactose and mannose, they are not epimers of each other. So let us have a look at this question. What kind of molecules are glucose and galactose? So here these are epimers of each other. The monosaccharides in aqueous conditions can occur in cyclic forms. So they must be having at least 5 or 6 carbons in their structure. So these monosaccharides, they undergo cyclization through internal hemiacetal or hemiketal formation. Let us have a look at this basis of cyclization among the monosaccharides. Here, when an aldehyde and alcohol react with each other to form hemiacetal, and the hemiacetal, when further reacts with another alcohol, acetal is formed. In case of keto compounds, when a keto compound reacts with another alcohol, the hemiketal formation occurs, whereas hemiketal further, when it reacts with another alcohol, ketal is formed. So in case of glucose, what is happening? The hydroxyl group of the fifth carbon acts as an alcohol and it reacts with the first carbon, resulting in formation of hemiacetal formation. So hemiacetal formation results in formation of alpha D glucopyranose, which is a which resembles a six-membered ring structure called as pyran, and so it is referred as pyranose. So this under cyclic form, the first carbon can have the alpha configuration or beta configuration. Now, how to identify the alpha or beta configuration? The carbon, if the hydroxyl group on the anomeric carbon is in same orientation to the hydroxyl group on the fourth carbon, the anomeric carbon is referred as alpha anomeric carbon. Whereas in case of beta anomeric carbon, the hydroxyl group is in opposite orientation to the hydroxyl group on the fourth carbon in the sugar. So this was about the alpha D glucopyranose and beta D glucopyranose. In case of uh, aldohexoses, the five-membered ring structures are also formed, which resemble the furan, which is a five-membered ring structure, and these are called as furanoses. But however, in this case, in case of aldosis, the six-membered aldopyranose ring is much more stable than the aldofuranose ring, and hence it predominates in the aqueous conditions. Only aldoses having 5 or more carbon atoms can form pyranose rings. Fructose, it under cyclic conditions, under cyclization, the fructose resembles 5-membered ring structure called as furan, and hence it is referred as alpha D fructofuranose or beta D fructofuranose. Here also the orientation of hydroxyl group on the anomeric carbon is to be compared with the orientation of hydroxyl group on the fourth carbon. Derivatives of monosaccharides. Several derivatives of monosaccharides also are found in nature. These can be glycosides, amino sugars, sugar acids, sugar alcohols, and so on. Glycosides, when hemiacetals of sugar react with an alcohol to form an acetal, it is known as glycoside. Glycosides are formed by condensation between the hydroxyl group of the anomeric carbon of a monosaccharide and the second compound that may or may not be a monosaccharide. If hemiacetal is glucose, the corresponding acetal is glucoside. If galactose is there, the corresponding acetal is galactoside. So here hemiacetal reacts with another alcohol to form acetal, and this is the basis of glycosides formation. Amino sugars. In amino sugars, a hydroxyl group is replaced by an amino or acetyl amino group. In glucosamine, galactosamine, and manosamine, the hydroxyl group on the second carbon of the parent compound is replaced with an amino group. So here is the structure of glucosamine and N-acetyl glucosamine. So you can see the hydroxyl group on the second carbon is replaced by amino group. Whereas amino group, if in turn it is attached to an acetyl group, it is referred as N-acetyl glucosamine. So here the anomeric carbon is in beta configuration, and hence it is referred as N-acetyl beta D glucosamine. Here also the anomeric carbon is in beta configuration, and hence it is referred as beta D glucosamine. In case of N-acetyl muramic acid, the N-acetyl glucosamine has a side chain or the lactic acid attached to the third carbon. So if lactic acid is attached to the third carbon to the N-acetyl glucosamine, it is referred as N-acetyl muramic acid. Other amino sugars are also shown over here, galactosamine and manosamine. These are some examples of other amino sugars. Acidic derivatives are also found among the monosaccharides. Both aldonic and uronic are found. So if the aldehyde group carbon is oxidized to corresponding carboxyl group, the acid is referred as aldonic acid. Whereas in the case of aldosis, the 6th carbon is, if it is oxidized to corresponding carboxyl group, it is referred as uronic acid. So these gluconic acids or glucuronic acids, in case of glucose, they can undergo cyclization, and the internal lactone formation can also occur in this case. So gluconic acid can undergo cyclization to form gluconolactone. Glucuronic acid structure is shown where 6 carbon is oxidized to carboxyl level. The carboxylic acid groups of the acidic sugar derivatives are ionized at pH 7, and the compounds are therefore correctly named as the carboxylates such as glucuronate, galacturonate, and so on. Sugar alcohols, the carbonyl groups in aldoses and ketoses can be reduced to the hydroxyl group to form sugar alcohols or alditols. Sugar alcohols, their name end with the suffix etol with the name of the parent sugar. For example, the glucose, in case of glucose, the sugar alcohol is called as sorbitol. In case of xylose, the corresponding alcohol is called as xylitol, and so on. Oligosaccharides, these are another kind of carbohydrates. These consist of short chain of monosaccharides which are linked to each other by glycosidic bond. Among these, disaccharides are most abundant, and the typical one is the sucrose, which is also referred as cane sugar, which is a disaccharide of glucose and fructose. In cells, most oligosaccharides consisting of 3 or more units, they do not occur as free entities but are joined to non-sugar molecules such as lipids or proteins to form glycoconjugates. Disaccharides examples are shown in this table. The examples include maltose, lactose, sucrose, trehalose. Maltose is the product of the starch hydrolysis by amylase and is composed of 2 glucose units. Lactose is found in milk, often referred as milk sugar. Sucrose is found in cane and beet juice. It is also found in sorghum and pineapple, carrot fruits. Trehalose is found in fungi and yeast, and as a compound found in the hemolymph of the insects. So here the condensation between the 2 sugar units results in glycosidic bond formation, and the condensation reaction occurs like the reaction that was shown earlier. The hemi one compound, one sugar unit, it acts as an hemiacetal, and another sugar unit, it acts as an alcohol. So the condensation reaction may result in formation of glycosidic bond. In case of maltose, 2 glucose units are linked to each other by alpha 1,4 linkage. So this is the disaccharide maltose consisting of 2 glucose units linked to each other by alpha 1,4 linkage. In case of lactose, galactose and glucose, they are linked to each other by beta 1,4 linkage. In case of sucrose, glucose and fructose, they are linked to each other by alpha 1 to beta linkage. So as there is no free aldehyde or keto group, this sucrose is a non-reducing sugar. Trehalose is also another non-reducing sugar in which the 2 glucose units are linked to each other by alpha 1 1 alpha linkage. Polysaccharides, these are the sugars containing more than 20 or so monosaccharide units, and some have hundreds or thousands of units. Some are linear chain, and the examples include cellulose, whereas others are branched one, which includes glycogen. Both glycogen and cellulose consist of recurring units of D-glucose, but they differ in the type of glycosidic bond that links the glucose units, and hence the different properties and biological roles are exhibited by these 2 polymers. Polysaccharides, these can be further classified into homopolysaccharides or heteropolysaccharides. Homopolysaccharides, they contain only one type of single repeating sugar unit, whereas heteropolysaccharides, they contain 2 or more different kind of repeating units. Homopolysaccharide is shown, so only one kind of repeating unit is there. It can be either linear or branched. In case of heteropolysaccharides, minimum 2 different types of repeating units must be there. So this is a linear heteropolysaccharide, and this is a branched heteropolysaccharide. Some homopolysaccharides serve as storage compounds, whereas some can have structural roles. For example, starch and glycogen, they have storage functions, whereas cellulose and chitin, they have structural functions. Cellulose is found in the cell wall of plants, whereas chitin is found in the exoskeleton of animals. Heteropolysaccharides provide extracellular support for organisms of all the kingdoms. For example, the rigid layer of bacterial cell envelope, the peptidoglycan, is composed in part of a heteropolysaccharide built from two alternating monosaccharide units. In animal tissues, the extracellular space is occupied by several types of heteropolysaccharides, which form a matrix that holds individual cells together and provides protection, shape, and support to cells, tissues, and organs. Here is an example of the polysaccharide starch that is shown over here. So you can see the alpha 1,4 linkage is present between the glucose units. So these are the glucose units which are linked to each other by alpha 1,4 linkage. The linear component is referred as amylose, whereas at branch points alpha 1,6 linkage exists and the branch component is referred as amylopectin. So two glucose units at the branch points are linked to each other by alpha 1,6 linkage. Glycogen is the animal storage compound and is often referred as animal starch. Like amylopectin, glycogen is a polymer of alpha 1,4 linked glucose units, but uh, the linkage at the branch points is also alpha 1,6 linkage, but glycogen is more branched. It is extensively branched compared to starch. So it on an average, every 8 to 12 residues a branch point can appear. Whereas in case of starch, the branch point appears after every 20 to 24 repeating units. Cellulose is a fibrous, tough, water insoluble substance which is found in the cell walls of plant, particularly in stalks, stems, trunks, and all woody portions of the plant body. Cellulose constitutes much of the mass of wood, and cotton is most pure cellulose. This molecule is a linear, unbranched homopolysaccharide consisting of 10,000 to 15,000 repeating glucose units. So here, in case of glucose, in case of cellulose, glucose units are having beta configuration. So the anomeric carbon is in beta configuration as compared to amylose and amylopectin and glycogen, which have glucose in alpha configuration. So anomeric carbon in these amylose and glycogen, they are in alpha configuration. This is the basic difference between the cellulose and starch or glycogen. So you can see the glucose units are linked to each other by beta 1,4 linkage in case of cellulose. So cellulose is a linear polymer. It is an unbranched homopolymer of glucose. Chitin is another homopolysaccharide. It is a linear homopolysaccharide composed of N-acetyl glucosamine residues linked to each other by beta 1,4 linkage. The only chemical difference between cellulose and chitin is the replacement of hydroxyl group at C2 carbon with an acetylated amino group. So this is the basic difference between cellulose and chitin, whereas the linkages between the repeating units is also same. Beta 1,4 linkage is found in both the cases. Chitin forms extended fibers similar to those of cellulose, and like cellulose cannot be digested by vertebrates, it is found in the exoskeleton of insects. So here is the cellulose that is shown, chitin, where N-acetyl glucosamine units are linked to each other by beta 1,4 linkage. Heteropolysaccharides, these include peptidoglycan, which is found in the cell wall of bacteria, which is a rigid component consisting of alternating beta 1,4 link N-acetyl glucosamine and N-acetyl uramic acid. So here the N-acetyl glucosamine and N-acetyl uramic acid residues are shown, which are linked to each other by beta 1,4 linkage, and this bond is the site of action by lysozyme. Aminoglycans are heteropolysaccharides which are found in the extracellular matrix, which is a gel-like fluid that is found in the animal tissues, between the animal tissues, and is often referred as ground substance. The extracellular matrix is composed of an interlocking meshwork of heteropolysaccharides and fibrous proteins such as collagen, elastin, fibronectin, and laminin. These heteropolysaccharides, the glycosaminoglycans, are family of linear polymers composed of repeating disaccharide units. So here some examples of glycosaminoglycans are shown. Hyaluronate is there, which is a heteropolysaccharide consisting of glucuronic acid and an acetyl glucosamine linked to each other by beta 1,3 linkage. Approximately 50,000 units, repeating units are present in this structure. So it is a very complex one, whereas other glycosaminoglycans, these are simpler compared to hyaluronate, and these include chondroitin sulfate. Here the two units, glucuronic acid and N-acetyl galactosamine 4-sulfate, they are linked to each other by beta 1,3 linkage. So there is one sulfate group in case of chondroitin sulfate, which is found in cartilage. Keratin sulfate, which is found in hair and nail. These are consisting of galactose and N-acetyl glucosamine 6-sulfate linked to each other by beta 1,4 linkage. So this is another example of glycosaminoglycan. Heparin is another one, which is consisting of glucuronic acid 2-sulfate, and the another is the glucosamine trio triple sulfate. 3 sulfates are present. Okay, so this is the compound which is having extensive negative charge, and it binds to the thrombin and is responsible for blood thinning. Thrombin is responsible for blood clotting, so it will not allow the blood to form by binding tightly to thrombin. Glycoconjugates are often found. Proteoglycans are macromolecules of the cell surface or extracellular matrix in which one or more glycosaminoglycan chains are joined covalently to a membrane protein or a secreted protein. Proteoglycans are major components of connective tissue such as cartilage, in which there are many non-covalent interactions with other proteoglycans, proteins, and glycosaminoglycans provide strength and resilience. So this is the example of proteoglycan in which the glycosaminoglycans may be attached to the serine of the protein through xylose, and xylose is a part of a oligosaccharide linkage between these glycosaminoglycans and the protein. So trisaccharide bridge is present in this case. So this is another example of proteoglycan. You can see the chondroitin sulfate, heparin sulfate, they are covalently linked to the protein. So let us have a look at the previous year questions. Which one of the following glycosaminoglycans is generally not covalently attached to protein as proteoglycans? So here hyaluronin is the one which is not covalently bound to the proteins, whereas others are the one which are covalently bound to the protein. So the correct example is hyaluronin. Glycoproteins are the one in which the sugar units are linked covalently to the proteins through either oxygen or amino nitrogen, and accordingly they are referred as O-linked glycoproteins or N-linked glycoproteins. So here you can see the sugar residues are attached to the proteins through oxygen, and so these are the O-linked glycoproteins, whereas if they are attached through amino nitrogen, they are referred as N-linked glycoproteins. Glycolipids are also found in nature. So these are the membrane lipids in which the hydrophilic head groups are oligosaccharides, which as in glycoproteins acts as specific sites for recognition by carbohydrate binding proteins. So here you can see the lipids, they are bound to the carbohydrate residues. So here the glycoproteins are glycolipids, they are found in the membranes of the organisms. So this was about the carbohydrates, and the major portion has been taken from Lehninger principles of biochemistry. So this was about the carbohydrates. Thank you and happy learning.